Sure, I
Welcome
can't.
everyone
Yeah.
to this week's edition of the Extracellular Vesicle Club. We're privileged to have Jennifer Byrne with us from the University of Sydney in Australia. And I have to say that today's topic is one that is kind of near and dear to my heart. I encountered some interesting issues with papers very early in my career. And today is also kind of an unfortunate day in that the president of Stanford has just resigned because of allegations about data manipulation. And so this is a very pressing issue, I think, for a lot of people around the globe. And you know, as a beginning microRNA researcher, I remember early on when I asked for some data from some microRNA microarrays, and I didn't get the data. And I asked again, and I asked several different groups about data, and I found that most groups that published in this area were not sharing their data, and that furthermore, when you got the data, sometimes you couldn't reproduce the results. But I never would have imagined that this went beyond, you know, mere maybe incompetence or lack of attention to detail, where we could have actually entire parts of the literature that are made out of whole cloth. So Jennifer, thank you so much for joining us today. And I'd like to invite you to take over the screen here and tell us about some of your findings.
Thank you, Ken. Yes, so thanks very much for the invitation. I'm going to be sort of presenting a bit of a mixed talk. I mean, some of this we'll be presenting some of our results, but there'll be quite a lot of focus just on a broad outline about paper mills and also what individuals can do, firstly, to, I suppose, protect themselves from these kinds of papers in the literature, but also to help with the much needed efforts of raising awareness about this kind of problem, particularly in the area of gene research. So I'm going to be talking a little bit initially about how we assessed trustworthiness of publications, either one paper at a time or many papers at a time when as may be relevant to literature from paper mills. I'll then give an outline about research paper mills and broadly what they are and what we think they do, talk a little bit about some of our results, and then talk a bit about reproducing experiments. I think that Ken has already alluded to that we can individually and collectively waste quite a lot of time attempting to reproduce experiments when those experiments may never have taken place. So I think that the era of paper mills forces us to rethink our approach to reproducing experiments in the laboratory, and then finish by a short consideration of what, you know, we can all do to improve the situation for our community. So I thought I'd start with this question, and I guess I just want you to have a think about this yourselves, but think about how many papers you read in a week and maybe extrapolate that to how many papers you read in a year. I started thinking about this when we wrote our most recent preprint, and people have been surveying researchers since the 1970s about their reading habits. And what surprised me about reading this literature is that we actually don't read that many papers. It's estimated that academics and scientists read between 150 and 400 papers a year. And that's not many, considering how many papers are published across the literature. So because we don't actually read that many papers, we should make each one of them count. We should think about what we read very carefully and think about what influences the papers that we do read. So this slide highlights, obviously, the the importance or lack of importance of the impact factor, and research has indicated that early career researchers are more likely to be influenced by impact factor in terms of choosing their reading, whereas more senior researchers, perhaps who are a bit older and more cynical, are less likely to be influenced by that. Either way, before you really start to invest in the published literature, it's worth investing some time to do some due diligence to work out whether what you're reading is what you think it is. So I'll talk a little bit about assessing research papers one paper at a time. Some of this might seem a little bit obvious, but the reason why I'm talking about what's trustworthy at an individual level is because this is essentially overturned when we start to think about paper mills. So if you're thinking about reading a paper, I mean, obviously, if you're reading a paper in your own field, you're probably fairly confident of what you're looking for in terms of trustworthy literature. But if you start to read a little bit outside your field, which I think we all do, Signs of trustworthiness are, fairly straightforward. I mean, trustworthy papers tend to investigate interesting topics, and they tend to also put forward specific and well-justified research questions that are based upon, a thorough consideration of the previous literature. They also feature appropriate and well-designed methods, and then clearly explain results that clearly align with the research question and the methods as they're written. The discussion addresses the results directly and, very importantly, recognises limitations of the study, because all studies, of course, have limitations, and the entire paper will be supported by a series of well-chosen and meaningful references. So this seems pretty obvious. So it's quite okay for research papers to have surprising results, and of course many research papers do describe results that are in some sense surprising, but research papers should appear to be plausible. And again, if you're an expert in your field, you're probably fairly well qualified to judge that. But if you're reading a little bit outside your field, that plausibility can be harder to assess. So here, if you're reading outside your field, it can be worthwhile putting papers in context with other literature that's been published to try and work out, well, what is really plausible in the field, or what's plausible in terms of the types of papers that have been published in the past. So I think where you should perhaps become a bit wary is if you see unexpected patterns in the literature, and these patterns might be described by unusual numbers of papers or concentrations of papers where these papers are highly similar. So they all kind of look like they're unusually similar. I'll give you an example in a minute. Papers where all the literature is coming from one country is also difficult to understand, unless, of course, the thing that you're studying is of particular interest to a single country. But that's pretty unusual in the field of, basic biology. If you start seeing papers that seem to be produced by the same authors that are very similar, that can be an example of salami slicing. So salami slicing is just a term for where authors are dividing their research into the smallest publishable units to derive the maximum number of publications from their work. But if you see very similar papers with different authors, this can be a sign of paper mills. I'll just tell you how we started to study this. About in 2015, 2016, I was a cancer researcher studying genes in cancer, and I noticed sort of an implausible concentration of 5 papers around a gene that I had cloned in the late 1990s, which is highlighted in yellow here. Now, almost nobody was studying that gene, including me. And I found these 5 papers that all referred to the gene, different cancer types, and with surrogates for the term proliferation in their title. So they were papers that were knocking down this particular gene and looking at these effects in cancer cell lines. So very simple experiments. I was surprised that there were 5 of these papers. I was even more surprised when I compared the figures. You can see here that these are 2 figures from different papers with unrelated authors. And the format of these figures is very, very similar. In some respects, I also found these results to be implausible, because you can see that knocking down a single gene in this particular cell line is really showing a very dramatic effect on proliferation or survival, and that seemed to me implausible as well from someone who had done these kinds of experiments. But I became most concerned when I realised that some of the nucleotide sequence reagents in these papers were not what they were described to be. So this paper shows the effect on expression of knocking down this particular gene with this reagent here. The only problem is the negative control reagent is identical to the knockdown reagent. So even though the authors are describing very different results, so no effect here, that's impossible because these sequences are the same. They're both targeting sequences for that particular gene. So I found that very unusual. I was starting to work with my colleague Cyril Labbé by this point, and Cyril wrote a tool called Seek Blastn, which is a semi-automated way of fact-checking nucleotide sequence reagents to mimic essentially what I was doing by hand, just using BLAST. Okay, so we'll come back to that a little bit later. So I guess right from the beginning, we suspected that the numbers of papers that we were finding, their similarities to each other, suggested some kind of external influence in the production of these papers. And we now talk about these things called research paper mills. Paper mills were probably, they've been described in the literature since perhaps about 2010, as far as I'm aware, in terms of, in the kind of setting that we're thinking about. We really don't know a lot about them because they are, of course, hidden organisations, and they sell undeclared research services. So the problem, of course, is that researchers don't declare the use of paper mills when they publish. It's, of course, fine to declare the use of an editing service or, you know, any kind of outsourced service for your research, as long as you declare that and make it really clear that you did obtain these services, you know, other than doing it yourself. So it's believed that paper mills probably sell a mixture of genuine, falsified or fabricated data sets and manuscripts. If we're talking about scaling production, fabrication is the easiest and cheapest way to proceed. It's pretty clear that they sell author positions on manuscripts. They can take care of manuscript submission, editorial handling. This probably happens either by placing associated editors in the position of overseeing special issues, and possibly by also paying editors in journals as well. They'll take care of peer review by nominating sympathetic peer reviewers if journals will allow that. They probably take care of post-publication commentary, for example, on the website PubPeer. And I think a growing number of people think that they're also forward-selling citations of their work as well. So there's certainly these kinds of advertisements, calls for author positions are very easily identified on the internet. This slide was given to me by my colleagues, Reese Richardson and Thomas Sturger. The paper mills probably work within an ecosystem that I've shown here in this sort of grey circle. And they work, you know, I was talking to a colleague of mine the other day, and she said to me, you know, the thing about paper mills is everybody gets what they want. And that's what makes them work. At least in the short term, people are getting what they want. So the wheel is largely driven by people who need papers and can't obtain them through their regular work. And those people give their identities as authors and also their money to the paper mills. And the mills produce products, which of course go to journals. So the journals benefit because they obtain a steady stream of manuscripts. They might be receiving dollars in terms of article publication fees and, of course, future citations to their work. The institutions then benefit, at least in the short term, through publication metrics that can be leveraged for future funding and prestige. And those institutions turn the wheel by offering career rewards for publications, sometimes even financial rewards, sometimes by leveraging publication quotas that people feel that they can't meet. So you can see that this wheel turns pretty efficiently. So we've talked about this in previous commentaries. This is a paper that we published in 2020 and more recently in Nucleic Acids Research. I'll just present some of our recent results that were published early last year. So we applied the Seek Blastn tool to, you know, about just under 12,000 papers. And this involved verifying, you know, many thousands of sequences across many thousands of papers, which was a pretty difficult job undertaken largely by Yasunori Park in my team. And Nori identified 712 papers that had wrong sequences, roughly 2 sequences per paper. These papers were being highly cited in many cases. And in the case of one journal, we found more than 10% of the original papers that described at least one wrong sequence. These are the 2 journals that we screened. We screened the journals Gene, published by Elsevier, and Oncology Reports, published by Spandidos. So Oncology Reports was the journal that mostly published more than 10% of papers with wrong sequences. So remember, these are all papers, not just papers that describe sequences. This is every original paper published across the journal. And the majority of these papers came from China, but there were certainly papers from many other countries as well. So more recently, in a preprint that we posted at the beginning of the year, my student, Pranajan Pathendra, he identified that up to 38% of another journal, Molecular Cancer, published papers with wrong sequences. And in 2020, when we did this, the impact factor of this journal in 2020 was, I think, 27.4. So we've identified 4 different nucleotide sequence error types. But the most recent one, I think that's probably in a sense the most interesting, is Pranu found that some circular RNA papers seem to describe non-verifiable circular RNAs. So we actually can't prove that the target exists through examining external databases. This is, I guess, a new development, and we're starting to study other kinds of non-verifiable reagents in publications as well. So we proposed right from the beginning that this might be reflecting a system of mass or industrial scale fraud. We might be seeing fraudulent or fabricated papers claiming to examine human genes, and that this content could be created using templates for speed and efficiency, but this rapid content generation could be associated with high error rates. Okay, so essentially when I think about the literature and research fraud, I think there's two types of literature. There's literature that's inherently resistant to fraud, at least at the moment, and research that's more vulnerable. So research is resistant when it's essentially about the same effort to fabricate data as it is to produce it through genuine research. But research becomes vulnerable when it's actually much easier to fabricate data than it is to conduct the genuine research. And I think most of us here, I'm sure most people here are experimental researchers, and you know that experimental science is difficult, it's expensive and slow. So research can become vulnerable to fraud when it's any of these things, but when it's all of these things, we can really end up being very vulnerable indeed. And in terms of my experience as a gene researcher, I know that this research is difficult, expensive and slow. I'm sure many of these steps apply to EV research as well. We've got to get funding, we need a regulatory approval, we've got to get hold of resources. Then we do the experiments, and that can take a really long time depending on how difficult they are, how many of them you do, how many people you have doing them. And then of course you write the manuscript. Now, if paper mills bypass the entire research process, they can just start by writing a manuscript, which puts them at a very significant advantage over genuine researchers. Gene research, I think, is also vulnerable because it offers what we've referred to as scaling and dilution factors. So if you're going to produce really large numbers of papers, I'm talking about thousands, we need a lot of topics, and of course human genes provide this because we have 40,000 genes and of course many more transcripts. They can be applied in different ways. You need to distribute these papers across multiple authors, otherwise it's going to look very unusual for low numbers of authors to be publishing so many papers. And of course, gene research can be done by many different types of people in different types of fields, institutions, countries. We need lots of journals. There are many journals that will publish this kind of work, and this can all go on for a long time if supply and demand continues, if it's not detected, and if we choose not to do anything about it. So these are all scaling factors that can be, I guess, harnessed by paper mills. So what does this come down to when you're looking at the literature? So I talked about trustworthy papers that are likely to be the result of individual efforts, but I think papers become less trustworthy when they show features of being scaled, and these features are listed to the right. So paper mill papers are often about topics that just aren't very interesting. It is really hard to understand why anyone was so interested to examine this particular gene in this particular context, using these techniques. The research questions that paper mills attack are generally, in terms of our experience, they're often very superficial and they're novelty based. And that's because, of course, you can scale novelty. You can say every single time that this hasn't been done and it's worth doing, and you don't really have to exert any more cognitive effort than that. So the methods tend to be very generic and increasingly superfluous. So it's as if the authors are trying to impress by a sheer amount of research as opposed to, the intelligence of that research. The results are often generic, they're almost always positive, and they're often implausible, as I showed at the beginning. The discussions are often generic. Again, there's very vague calls for more research, the need for more research, combined with frequently overstated significance, such as, this kind of thing could be informing some new therapy for cancer. And finally, the references are often poorly chosen, and we suspect increasingly the papers from mills probably cite papers from other mills, and that's one way that they can achieve these kind of high citation numbers. So if we're thinking about reproducing experiments upon a background of literature where there might be paper mill papers within that literature, I think it's really important before you start doing experiments to do what you can, through in silico approaches or through data triangulation approaches first. So try and look for evidence in the literature to support what this paper is saying. And I think it's a good idea to prioritize data from high throughput experiments if you can, because of course we all know that the literature is full of publication bias, positive publication bias, which of course paper mills exploit as well. You can do things like, checking the identities of nucleotide sequences. You can look at cell lines and see whether any of them are contaminated or misidentified. So do this kind of due diligence. And if you can't really do that, then think really carefully about the results. Do they seem realistic? Do they seem plausible? If you're not sure, ask somebody else, ask someone who might be closer to the field than yourself. And then if you're designing reproducibility experiments, I think it's really important to think about the minimum piece of data that you want to reproduce to have confidence in these results. And try to match the experimental conditions as closely as possible. I know within my lab, I remember I had somebody trying to reproduce some experiments, and she did those experiments, they didn't work, but we weren't doing it in the same cell line that the authors were using, And so then when it doesn't reproduce, you can say, oh, well, it wasn't the right cell line. So it's probably best to go to the effort of matching the cell line from the beginning, or doing something else where you're really trying to match the experimental conditions as closely as possible, because otherwise you can tell yourself, oh, well, I just need to do that in another cell line, and essentially you're just wasting more time. I think it's very important to define hard stop points, and by that I mean, if this doesn't reproduce, I'm leaving this paper, and stick to those, because I think we all know we can end up in these loops of just trying that one more experiment. So as I mentioned, we've got this tool, the Seek Blastn tool, so you could use that. It's freely available online. You can upload papers and check sequences. We've published as well a very extensive standard operating procedure telling people how to manually verify results from Seek Blastn. And that includes a discussion about verifying particularly RT-PCR primers for MERS, which have got different targeting requirements, particularly in terms of the reverse primers. And in our most recent preprint, we also described verification procedures for either divergent primers that target circular RNAs that basically face differently on transcripts, and also the requirements for single circular RNA reagents, which need to target the backsplice junction sequence in order to have specificity. So this is all online as well. So I think, yes, we really have to sort of change our minds around reproducibility of the literature to thinking, from thinking when something doesn't reproduce that it's our fault, to actually thinking, no, maybe it's that paper. Maybe it's not me, maybe it's you. So I'm pretty much at the end now. I just wanted to also ask you whether you've ever received this kind of training or advice before. I gave a similar talk in France last week, and when I asked a room of about 100 people, only 2 people put up their hands. So it seems as if this kind of discussion isn't really taking place in the biomedical community, and I think it's quite urgent that we have these kinds of discussions and these kinds of, I guess, semi-education sessions. So perhaps what I want to leave you with is thinking about what you can do at your institution, what we can all do. I think it's really important to start conversations around publication integrity and paper mills within individual laboratories and groups. You can ask for publication integrity training to be included in any research integrity training that you might need to undertake. That's frequently a mandatory requirement at universities, particularly for students. Everybody's, certainly at my institution at the University of Sydney, all 4th year students undertake research integrity training. We've only just recently started to introduce an element of publication integrity into that. And when I mean publication integrity, I'm not talking about authorship and things like that around, manuscript preparation. I'm talking about how do you actually assess the literature. If you're doing journal clubs, you can also include a focus on publication integrity in those. You can give a presentation to your own team or to your own institution, and I'm happy to share the slides from this presentation today. I think that paper mills are probably affecting the EV field. That's not something that we've examined directly, but we have identified EV papers with wrong sequences. The EV field has got, these kind of same scaling and dilution factors that I talked about. So I think it's just really important that we're all aware of what we're reading. So finally, just as parting words, if you're the expert, I think trust your own expertise. If you're not an expert in a field, seek advice when you're reading the literature for the first time. Read in depth, particularly where you think that you might build on the results. Again, trust your own instincts. If you think that something's wrong, or if something looks too good to be true, or if there's something odd about a paper, then there could well be, because trust in the literature needs to be earned. So I'm just going to thank the members of my team, and this is information if you want to learn more about what we do at the University of Sydney. This is our website, and just want to thank you all very much for attending, and I'll be very happy to answer any questions and to have some discussion now about this topic. Thank you.
Fantastic. Well, thanks so much, Jenny, for this great overview of what's going on around the world in research fraud. I think it's just so shocking to many scientists to realize that there aren't just issues with with poor experimental design or with incorrect interpretation, but there's actually these systematic, well orchestrated attempts to subvert the literature. And indeed, I teach a research like a reproducibility and research course at my institution, and we specifically don't cover the topic of fraud in that because we see that as being just so beyond the pale. But I think you make a good point that it is important to educate people that there is the opportunity for this. So before I just want to remind everyone that if you have any comments or questions, please put them in the chat box. I'm going to allow unmuting in just a minute here, so that you can speak with Jennifer about what she's had to say, maybe respond to some of the questions that she had for you. But first, I would like to share my screen for just a second. And I'm going to show you a slide that I have presented at a few EV meetings recently, but that it might be worth to take a look at here. All right, so this is a slide that contains some images, and I just want to emphasize I'm not accusing anybody of doing anything wrong here specifically. These are just some examples that I have pulled generically from the literature that kind of illustrate some of the concepts and some of the features that we might actually observe in paper mill papers, or at least in papers that have perhaps cut some corners in the EV literature. The MICEV guidelines, we have these guidelines in the field that have been established by the International Society for Extracellular Vesicles, first in 2014, then in 2018. So these minimal information for studies of EVs currently being renewed, and we hope to have this renewal of the guidelines document published later this year. But unfortunately, sometimes when you put guidelines out there, they also become a kind of template for people to, we think we have to just satisfy these 3 criteria and then we can publish our EV paper. And so one of those things is showing images. And so again, I'm not saying that this image is necessarily not of EVs. It could just be a bad EM image. But, we see a lot of these things in the literature where it seems like somebody feels they have to show an electron micrograph, and they show something that, frankly, we don't know what it is. Like, where did this come from? What is it actually? What is actually being depicted here? Because are these things really showing the morphology that we expect of an extracellular vesicle? So that's a question. And then we come to, the second thing that for the last decade, everybody has thought that they have to put a nanoparticle tracking analysis graph in here. What is the size distribution of our EVs? What does this actually mean? Well, it's very interesting. If you look at the concentration here, particles per mil on the y-axis, are they really able to detect 7 particles per mil? That's impossible. So what is this axis really? We don't know. And when we look over here, we actually see that there are some overhangs. How can you have an overhang on a size graph? And to me, it appears that somebody just took a pen and then maybe a highlighter, a red highlighter, and just highlighted this thing, just drew this by hand, perhaps. We also see some sloppy things. I think over the years, we've kind of assumed that lipid dyes are going to let us know whether an EV has been taken up by a cell. And then finally, we have these characterization experiments where MICEV says you have to look at several transmembrane proteins. And you also want to look at something that you think is going to be, maybe inside the EV, perhaps a negative marker, something that is depleted. And when we observe these bands, though, what we see is that first of all, we don't see more than one condition represented or more than one repeat represented. And we also see some very curious things. So those of you who have done CD63 Western blots, you know that typically what you will observe is a smear. So something that's very, very broad, not a sharp band. And this isn't because there's anything wrong with the antibody. It's because CD63 is heavily glycosylated as a transmembrane protein. And so we see a smear in most of our experiments. It's very unusual to see a sharp band like this. And yet you can find thousands of papers in the literature where they're showing a sharp band for CD63. And so I would just say that, overall, it seems like there may be hundreds, if not thousands, of papers that have entered the literature, and what they are marked by is, first of all, they call everything an exosome. And in many cases, they're combining this quote unquote exosomal study with something that is also hot. So a microRNA, a link RNA, a circular RNA. So all of these non-coding RNA studies. And so this is, for me, when I see a title that follows this template, it tells me that this is a paper that I should be very careful about. So you see something like exosomal microRNA 3824 regulates protein Y in the link RNA, circRNA, whatever signaling pathway Z axis. When I see the word axis in a title, that tells me right there that it's probably a paper mill paper. Not to say that every paper with axis in the title is fraudulent. I'm not saying that at all. And in fact, the reason that this is used is probably because back in 2008 or 2009, somebody did publish a great paper that was real on this topic and had a lot of influence. And so everyone wants to mimic this. But just I just wanted to share this as some of the hallmarks that I see, or that can be present in difficult papers in the literature. So anyway, that's where we are with that. So Jennifer, I want to ask you about your experience. You have generated some excellent tools to identify, especially these gene sequences that are incorrect. So what has happened with these findings? Do you find that journal editors are generally very receptive to discussing these matters, or do you rather think that the journals are more of a problem than a solution right now?
Yeah, Ken, that's a great question. Yeah, we have had a fair bit of experience in describing our findings to journals, and that's been, with some exceptions, a largely fruitless exercise. So at the end of last year, we went back and looked at the number of papers that had actually been either corrected or retracted, out of the 712 that we described. So some of the papers, when we described them, had already been retracted because we used them as kind of cases to build further, corpuses on. But I think of the papers that we reported, so we described these papers mid-2021, so about 18 months later, only maybe 2 to 3% of papers had actually been addressed. And most of those corrections or retractions seem to have nothing to do with our reports. They seem to be in response to things that were happening already, like image concerns or something. So yeah, a lot of journals are, they essentially do almost nothing. I should say where we've had success, it's been largely working with individual people. So individual people at publishers who are very motivated, who are very concerned about this issue, have been extraordinarily helpful, but those people are in very small minorities. And what often happens in publishing is people are in roles for short periods of time. So they'll be in a role, they'll be very motivated and do something for us, and then, for a variety of circumstances, they'll change roles, and then we lose that contact and we lose that ability to work with either a journal or a publisher. So yeah, it's really not working. I think that's one of the biggest problems that we face. Post-publication corrections are just simply not working at the moment. I saw that you put the link for PubPeer in the chat, and that's a very important tool. I do really advise everybody to download that PubPeer plugin, because that will flag everything that you read as either having a PubPeer comment or not. Of course, then you have to go to PubPeer and work out what that comment is. But that's a good idea, particularly if you're thinking about following up a paper. If you see that there's a comment on PubPeer about a particular paper, it's definitely worth having a look at that comment and thinking about what that could mean. But yeah, post-publication correction needs a lot of work. We've proposed some ways of doing that. I think we have to sort of lower the bar to correcting papers. We currently think of this as a bad thing, that papers need corrections or modifications, but really it should just be part of the life cycle of publishing, that things are not published and then set in stone, that they're subject to modification. And we need other ways of doing that beyond working with journals, because the journals, I think, are not motivated to do that. It's not in their business model. They don't earn money when they correct the literature. They only earn money when they publish at the moment.
Yeah, indeed. And that was my experience too. When I was, I mentioned that early in my time as a PI, I found a lot of issues in the microarray literature. And in my experience, when something has been corrected or retracted, it has almost always been because the authors saw that as the right thing to do. And they were the ones who insisted on it.
Yeah.
And only very rarely do you have somebody at a journal. And I remember there was somebody at PLOS One for a while who was really looking into these things, maybe not super actively, or maybe not with a lot of effect because there was just so much volume. But over time, she made sure that the issues were brought to the attention of the correct authorities and looked into at least. But that's, as you say, it's very much dependent on individuals, and it doesn't seem that for most publishers there is a good workflow in place to ensure that these investigations proceed in a fair but expedient manner. Yeah, that's
right.
So I'd like to just open this up and encourage our participants today to weigh in on any experiences that they've had with this issue. And also to ask Jennifer about anything that might be on your mind. What about the authors, Jenny? Have you had anybody reach out to you and say, "Oh, wow, I never noticed that we put that primer in there twice"?
Yeah, occasionally we do. We have had people. I think in our most recent paper, I think we might have had one paper who wrote back and said, "Oh my God, yeah, we got those primers wrong. We're sorry," and a correction was published very quickly. I think you're right. If authors are happy to have their work corrected or retracted, then journals, of course, will work with them in a very expedient way. I think it becomes tricky when either authors don't respond, or what we have also seen is, we published this in 2021, we've also seen very similar responses from authors about issues around sequences, and identical sequences being put forward across numbers of papers to correct particularly these non-targeting sequences and knockdown experiments that are targeting. So we'll find that authors come back with almost the same sequence over and over to replace this particular sequence that was wrong. Now that's of course possible, but it seems a little unlikely. And we have thought that that could indicate that, again, a third party might be directing those kinds of corrections. We've even had corrections that have been incorrect. So people putting forward sequences to correct wrong sequences when those sequences are still wrong. And then we end up with published corrections that are wrong. And, this can be very destructive, really. So then we start to try to correct the correction. You can see that, as a small team, you can very rapidly disappear down a rabbit hole of continuing corrections. It's ridiculous. So that makes us suspect that the paper mills may be sometimes directing these corrections, which can confound attempts to actually truly correct the literature.
They're on a subscription service for any future responses.
That's it. Yeah, it's a bit worrying.
Sina, I see you have your hand up. Go ahead, please.
Thanks, Ken. Thank you, Jennifer, for a wonderful presentation. So one of the concern I had while reviewing some of the papers or reading through is we study long non-coding RNA cyst, which is only expressed in female cells. And then there are papers publishing, we looked at patient samples from male and we see these things, or in a male cell line. And then, based on their results, they would say there is this pathway being altered, and we are dinged by reviewers in our grant or whatever, oh, this has already been studied or whatever. But that's irrespective of that. How can, people reviewing it are not paying attention? Is this a female specific thing? And then there are papers on cyst. So something to, we have encountered, and I thought I should bring that up. Thank you for this wonderful discussion. It's always hard to distinguish what do we believe and what do we don't believe, kind of.
Yeah, that's a big problem, isn't it? You know, I know what you mean. So, you know, I think I've seen, yeah, it's pretty worrying. So I suppose when you see those papers that are talking about the expression of that gene in males, I mean, is that something, have you thought about writing a letter to an editor or writing that up? Because I think we've got to counter these kinds of claims in the literature.
When I'm reviewing manuscript, I bring that up with the editors, but like if it's already published, you know, sometimes I haven't done much. I admit maybe that's something I should actively make it a point to write to the editor and like flag it at least, because it gets propagated, like, you know, people cite those papers again and again. So
yes,
and when writing my own grants now, I've started putting, you know, there is this particular paper where they cite this thing, but it is on mail, so please do not ding up. It's almost like proactively protecting yourself because there is this false thing that is being propagated. So
yeah, I know. It's a huge issue. I mean, I've encountered that as a grant reviewer myself, where I remember last year I reviewed a grant where the authors were going to study a particular gene in a particular setting, and there was two bodies of literature, one that seemed to indicate that the gene did one thing and another where the gene seemed like it was doing something very different. And I looked at the very different body and thought, yeah, that looks a little suspicious. And the authors hadn't cited that other body of literature. But then the question in my mind as a reviewer is, have they not cited those papers because they suspect that they might be from a mill, or have they not cited them because it doesn't fit with their hypothesis? You know, so it creates this cloud of uncertainty, I think, and it makes it really hard to, it's just a layer of uncertainty that we don't need in science. Science is hard enough, you know.
yeah, thank you.
Yeah. But I think, you know, if this is something that's continuing in the literature, it might be worth thinking about writing a short article or something to alert people to that. And then you could cite that in your own grant applications. But yeah, it's very annoying.
So we have a comment in the chat box here, and I'm going to ask Mark to, if you're able to unmute yourself and ask this, because Mark is somebody, Mark Halushka, who has some experience with writing letters to the editor, as do I. And in trying to correct some of the literature, but Mark, you have a question about how we can alter this.
Yeah, I thank you. It's a great presentation, and you so nicely demonstrate how everybody has something to gain from this system. And I'm just wondering, like, what's the weakest link in there? What mechanism, or what are those four pieces between the authors, the mills, the publishers, and the institutions are the ones that would be most responsive to ending this practice, and what mechanism might work for it?
Mark, that's an excellent question. And I think that there's probably not an easy answer. I tend to focus a lot on the journals, because without journals, you know, paper mills really have nowhere to go. They need journals, and increasingly it seems they are looking to publish in more and more sort of prominent journals, or at least journals with higher impact factors. And some of those impact factors have probably been inflated through the use of mills. But, you know, I feel an effective mechanism would be more oversight over academic publishing. I mean, journals and publishers have tremendous power to do pretty much whatever they want. I would love to see an external oversight over journals and publishers that would provide opportunities for journals to do something effective against paper mills, and if they don't, they are simply closed down. I think we have too many journals at the moment with not enough content to publish. I think a very effective approach would be to very, very swiftly act on paper mill papers when they're published. So immediately flag them, and if authors don't respond to concerns, you know, either issue expressions of concern or retraction so that these public papers are highlighted in the literature. I mean, that's going to protect the researchers who are currently using these papers. In terms of actually stopping the production, you know, this is obviously an issue of academic incentives, and the thing about incentives is, people have been talking about trying to realign incentives in academia for a long time. So firstly, it's not an easy thing to do. And secondly, when you change the rules, it seems as if it takes a long time for academics to catch up. So, people don't change their habits overnight. So that's a bit of a roundabout answer, but I would like to see a lot more oversight over academic publishing. And I think we need to reward journals that do the right thing and punish those that do the wrong thing.
I often think about money. Big money kind of person. I'm in academia, but those who control the purse strings also have the power to make changes. And so if you look at what is done to regulate financial institutions, for example, what do we have in the research on the research side that is similar to that? We have funding agencies, and they review the grants and they make sure that people are submitting their reports and so on. But I think that there is an opportunity, a little bit more opportunity for regulation, not just self-regulation of our community, but maybe these agencies, these institutions that have experience with supervising, with funding, with administrating, they could perhaps take a larger role in this area. We already have mechanisms for evaluating journals, right? How does a journal get listed in PubMed? What does it mean to be in PubMed versus PubMed Central, and so on? But perhaps there's an opportunity to make that a bit more stringent and to make sure that there's, actually a very good control, I should say, of the profit motive here, because publishing is a very lucrative business as well.
I think that's right.
And there's no incentive, right? Unless you're going to be cut off from that information source or from that dissemination source, there's no incentive for a publisher to say, I don't want this paper.
Yeah, I think that's right. I'd like to see, journals evaluated by not only what they publish, but what they correct. And if we had a system where journals were judged based upon both the speed and, the speed and the extent to which they correct problematic literature, then we could have, for example, funding agencies requiring their grant recipients to publish in those kinds of journals, journals that actually have standards around post-publication correction.
Excellent point. Susan, I really like your question because this is on a lot of minds right now. Go ahead.
I've been trying to think about AI and the goods and the bads. And it occurred to me that it could, it can be used for good or for ill, but in this particular field, and I don't really understand it very well, to start with that, but in this particular field, it seems to me that sort of, like what you did in your lab, Jennifer, could potentially be done by AI in a way. Is there a better way to do this somehow with AI? But also, is there a way to protect against the AI being evil?
Yes, that's a really good question. And a lot of people, I think, are thinking about this at the moment. I think you're right that, AI is certainly already being used by people to, do, more effective, broader scale literature searches. There's obviously many ways that AI is going to be used in research in the future, and it's being used now. But we are worried that AI will increase the capability of paper mills to produce increasingly sophisticated and plausible manuscripts. So I think that possibility, one of the positives of that possibility is I think that has kind of galvanized people's attention towards this problem in the last few months, particularly since ChatGPT was online. There's obviously a lot of concern or discussion in education circles about the use of, models, tools like ChatGPT in terms of creating content that students will submit as their own work. So, in terms of helping or hurting, I think it's definitely both. It seems as if AI is going to progress very rapidly, and I think that's one thing that does concern a lot of people. The speed with which these capacities are gaining power means that people are struggling to catch up and respond in an effective way. So I suppose we're saying, you know, if we know we have a problem now, we really have to get on top of that now, because in a few months or in a year's time, the problem could be looking very different.
Thank you.
Yeah, no, thank you.
Any other questions for Jenny here before we wrap up? Comments? You know, one thing that troubles me a lot about this debate is the very unfortunate, let's call it geopolitical alignments here. And, you know, so we certainly don't want to pick on researchers from any one country, region, any one block of geopolitical alignment, whatever. And yet we do see that there are differences, you know, especially I think in the paper mill area. How, as you know, as somebody who is very familiar with this, do you approach that question so that there's not a chance for someone to say, oh, this is a racist comment, this is a, you know, this is an unfair discrimination against a particular country or region? You know, how do we be sensitive and inclusive while at the same time advancing research integrity?
Yeah, that's a great question again, Ken. I think we do this by, we never use the features of authors to screen papers. We always, you know, for example, if we're looking at a journal, we look at every paper in that journal. So we're not looking for papers from individual countries. One thing I think to add to the AI discussion, one of the things that worries me about AI is if paper mill papers at the moment, I think we've all recognized that they kind of deviate from best practice in many respects, but I think AI is going to close that gap quite rapidly. One of the things that I really worry about is if paper mill papers become very plausible, you know, almost undetectable, I think more and more people are going to be tempted to use them. You could imagine that somebody's going up for promotion. They know they need a couple more papers. They know now that there's a way of getting them, you know? So I think it could really broaden the market for these kinds of papers, and that really worries me. So I think it's quite dangerous to think of this as, you know, a geopolitically, you know, restricted issue. I think it's going to become an issue that will affect more people in more countries as paper mill papers become more plausible and more sophisticated.
Well, on that point, we will end today's EV Club. Actually, we will not. We will not end it. We have one comment here. Andy, you raised your hand.
Thanks very much, Ken. Thanks, Jennifer, for a great presentation. I've just got into the office, so it really helped my commute this morning. I think the AI thing is really, it's quite scary, especially, you know, I think as academics, we're focused on the sort of text-based AI issues, but for those who've seen the new Indiana Jones movie, you know that AI can be used to sort of enhance images and things. And I think I'm concerned about scientific images being compromised by the use of AI tools. And I think that's probably going to be even harder to detect. So I'm the editor-in-chief of one of the ICEB journals, and I think about how can we there's some ways we can get authors to authenticate their scientific data as they're submitting them to journals. Is this something we need to start thinking about? Because I think there's so much trust involved in the publication process, and it hasn't really changed that much as new technologies have developed. So I was just wondering what your thoughts might be on, how, and I know some journals have quite sort of lengthy forms. You have to supply original data. That's not consistent across journals. And I have an example of one paper I reviewed for a very high profile journal where the data was supplied. I identified several issues with that data that I alerted the editor-in-chief to. I never heard back. The paper then appeared in another high profile journal as it was. and you can't, the original data wasn't supplied as supplementary. So there's all these different issues. So I'm wondering what your thoughts are on sort of how we can authenticate data that could help with this process.
Yeah, that's a good question as well. I think for the moment, from what I understand, AI generated images can be identified through, because of how they're actually constructed. You know, there's kind of, I don't know how to describe this, but there's often sort of patterns within, if you sort of unpick them, there's patterns in those images that indicate that they were deliberately constructed as opposed to just captured. So that's a feature that exists at the moment, but that's probably going to disappear over the coming months. Again, there's no really easy answer and a quick response, but again, to encourage people to really look at the data and the plausibility of those data. You know, I think that can be a giveaway. You can create a beautiful image, but it may not make a lot of scientific sense. And by thinking about, you know, your understanding of the field, really, are those data supported by the literature? Of course, this becomes more complicated when, you know, you've got more invented stuff in the literature, but still a lot of that stuff might be considered to be going in one direction or particularly random or something. So I think, again, looking at the data and what it means. But yeah, you're right. It's a huge issue.
Yeah, thanks so much, Andy, for your comment there and your perspective as a journal editor too, because I know that these issues are constantly on your mind because you want to have the highest quality research possible that's published in the Journal of Extracellular Biology. So thanks for being one of the gatekeepers for our field. Good. All right. Well, I'd like to thank everybody for joining today, but especially Jennifer, thank you for your insights, for sharing your experience and expertise with us. And I think all of us have some things to look out for as we read those papers that we have time for, as we select the ones that we want to invest our efforts into, and as we think about how we can maximize the contributions that everybody is making to scientific research around the world. So thanks so much. And take care, everybody. Hope to see you again soon at a future EV Club. Bye now.